Hello everyone and welcome to the next video here going into the nervous system. So in the last video we talked about the structure of a neuron, and in this video now we're going to talk about how those neurons communicate via something called an action potential. So I switched to a white background, I know, a little brighter here, just to better match the colors I use in what we're going to go through today when diagramming the action potential. So we're going to look at how we generate what's called a resting membrane potential, that we can use that potential energy to then use what's called the electrochemical gradient to send signals very, very quickly down the neurons. And so we're going to talk about how those initiate and then travel down and propagate down neurons to the terminal. So here, we're just going to be working through this as we go through. First step here is building resting membrane potential. So here we have cell membrane. So of course, cell membrane would have phospholipid bilayers lined up in it. So that's what these lines represent here. And then I have a few proteins embedded in this membrane that we're going to talk about. Here we have the ECF, which is the extracellular fluid, and the intracellular fluid is right here. All right, the first protein channel we have here is called the sodium-potassium pump. So this pump, what it does is that it loads in 3 sodium ions and pumps 3 sodium ions out of the cell. And when it does that, it brings 2 potassium ions into the cell. And while doing this, it does use ATP, hydrolyzing it to ADP in order to power this pump, because it's pumping things against their concentration gradient, the direction they don't want to go. All right. So when this pump is going, think about what it's doing. It's building. So when I'm drawing out here today, sodium will always be in red, potassium will be in blue. And brackets mean concentration. So what ends up happening, the concentration of sodium outside the cell is high, and the concentration of sodium inside the cell becomes low. So that builds a concentration gradient, and every time this pump goes, you're going from an area of low concentration to an area of high concentration. So that's why energy is needed. Also, if we look at potassium, we're removing it from outside the cell. So the concentration of potassium will be low outside, and the concentration of potassium will be high inside. So that's the chemical gradient that we build. So chemical gradient is the first thing we have. But now think of terms of charges. Charge-wise, we're pumping 3 positive ions out of the cell, and you're bringing only 2 positive ions back into the cell. Let's imagine, negative charge proteins, chloride ions, and things like that. They aren't moving in this situation. And we're only moving 3 positives out, but you're only bringing 2 positives in. That's an unequal charge distribution. So what ends up happening is that you end up building a positive charge outside the cell, and you start building up a negative charge inside the cell. You get an electrical difference. And what we're forming here is in what's known as the electrochemical gradient. So electrochemical gradient here. And so that's a combination of the electrical difference and the chemical difference inside and outside the cell. The electrical difference can be measured with a voltmeter as well. And so at rest here, we're usually at minus 100 millivolts. So the millivolts are the voltage used, the designation for the units here for that voltage. So gets about minus 100 millivolts with this pump going. But it doesn't usually get more than that, because there's a channel here called a potassium leakage channel. So this is a channel protein that's always open, and potassium can leak through it. Now, things want to move down their concentration gradients, and opposite charges attract. So if a potassium ion is inside the cell right here, it's in a negative environment, which it likes, but it's in a high concentration, which it doesn't like. So this leakage channel has some positive sodium ions leak out, but at the same time, some also leak back in. And so here, when potassium leaks, what happens is that the resting membrane potential, or the voltage, gets down to minus 90 millivolts. And we talked about this back in one of the early videos for generating a resting membrane potential in a cell, where these potassium channels are the key in helping maintain most cells at this minus 90 millivolts. However, in neurons, we have an additional channel here called a sodium leakage channel. This sodium leakage channel now allows sodium to move in and out. And if a sodium ion is on the outside here, it's in a high concentration and it's in a positive charge when it's a positive charge ion. So it's unhappy. And there are not a lot of these sodium leakage channels, or you will automatically depolarize a cell. So in this case now, sodium will leak in, and some can leak back out, but most will leak in, and it brings neurons down to minus 70 millivolts. And then the thing is, as some leaks in, you'll get the potassium pump over here, which goes again. And then you also get these potassium leakage channels also keeping this resting membrane potential right around minus 70. Now, if you change from this minus 70, say you go from something like 0 millivolts to minus 70 millivolts, that is called polarizing. So polarizing. Or if you go from minus 70 to 0 millivolts, that is called depolarizing. So making something polar or nonpolar means it has a sidedness to it. That's polarities. Going from 0 to minus 70, you're increasing the difference between the two, whereas going from minus 70 to 0, you're decreasing that difference. You're depolarizing. There's also a term called hyperpolarizing, which goes up to like minus 110. Repolarizing is resetting it. So it's important to have these terms down before we move forward here. Alrighty, so now let's move forward and actually diagram how an action potential works. So this cell is sitting at that minus 70 millivolt resting membrane potential. And it's there, it's sitting, it's waiting, it's good to go. Now, the purpose of a neuron is to send a signal very fast when it hits certain conditions. This condition is called threshold. This threshold happens to be at minus 55 millivolts. So if this neuron depolarizes enough to reach minus 55, so here it would be depolarizing, it reaches minus 55, threshold was reached, and now these particular channels called voltage-gated sodium channels will open and you fire an action potential, and there's no stopping it at that point. So that's why this minus 55 is minus 50, minus 55. It can be a range right there, is so important. So what can get us there, or what can get us further away from there? Well, of course, you could have positive ions coming into the cell, anything like sodium, potassium, calcium, or you can have negative ions going out of the cell. Typically, chloride is a negative ion when referring to negative ions. So that would cause depolarize. The opposite could happen. So that could then this could lead to that minus 55. But what if you have positive ions exiting the cell or negative ions coming into the cell? In that case, that would lead to more polarization. It would be an inhibitory effect, and that could be hyperpolarization. That would bring us further away from minus 55. So if we look down here, here is the diagram of an action potential. So here I have those depolarizations and further polarizations happening on this graph. You can see minus 75 here. This is where resting membrane potential is, and we see it going above and closer to this threshold line right here, or going further down. And then eventually right here, we reach that threshold point at minus 55, and then you see an action potential start. So it all depends on what the stimulus is, what the receptor is, and so forth that leads to either of these events. But let's say we now have enough, say it's salt, and you have salt receptors for taste, and you have sodium ions coming into the cell, and you reach this minus 55 threshold here. So you have enough ions coming in from this sodium binding out here or something like that, and you open these channels here. They're called voltage-gated sodium channels. Now, voltage-gated sodium channels, they're typically closed. They have these gates here. And when they hit minus 55, they go through this magic thing where they open those gates. And because you have a very high positive charge outside and that negative charge inside, and a high concentration of sodium outside the cell, because we're looking at what we built for resting membrane potential, sodium will rush into the cell and it rushes in so fast that it goes all the way up. So sodium comes in, it goes all the way up to plus 30 millivolts. So here you see that happening down here where you hit threshold. So sodium comes in and you fly all the way up to plus 30 millivolts right here. So again, this is a depolarization right here when that happens as well. And you have initiated an action potential, and this will continue down the neuron until it reaches the end, opening the next voltage gated channel and the next and the next. But why does it keep going this way and not turn around and go backwards the wrong direction? Well, these voltage gated sodium channels inactivate right here. So this one is now inactive. There's this little ball that comes in and blocks it. And so sodium can't come in or go out in this case, or reopen. And that makes it important so that that section of the neuron can reset and the action potential will continue unidirectionally down that particular neuron. But now, how do we reset that area and get it back to that resting membrane potential? That is where voltage gated, so V gated is voltage gated potassium channel comes in. So these channels then open at that plus 30 millivolts. And if you remember, we have a high concentration of potassium inside the cell. And now we have a positive environment inside the cell. So potassium is very unhappy. So potassium will fly out of the cell. And so that's a positive ion leaving the cell. And it can go all the way to like minus 110 millivolts. So much potassium flies out. And so that's repolarization and potassium out. So here we see that happening down here. So here potassium leaves or potassium out, and this is repolarization. And you see it goes all the way down here to like minus 110, and it goes well below resting membrane potential. Then eventually between the sodium-potassium pump and the potassium leakage channels, it can reset back to resting. But so then that can come all the way back then to resting membrane potential, and it's ready to fire again. Now, the thing is now, when can you fire another action potential? When can this section fire again? If you still have sodium coming in right here, if there's still sodium coming in, you're going to fire another action potential. You're going to send another stimulus. And this is resets in within 5 milliseconds, but there's a point in which you can fire and a point in which you cannot fire no matter what you do. So here, from the moment threshold opens to during the reset phase right here, this is called the absolute refractory period, meaning you absolutely cannot fire an action potential during this period. Because that is when the sodium is inactivated. It doesn't inactivate until it starts resetting here and the charge starts going back down. Once that sodium channel reactivates, you can then fire another action potential. But then it's down here at minus 110. So to have enough stimulus come in to reach the minus 55 again takes a substantial amount of stimulus. So between there and when you get back to resting, so between that point right there pretty much and over here, so this little box right here, this is called the relative refractory period. So the relative refractory period means it's relatively difficult to fire another action potential because now it has to go from here all the way to there. So it has to be a very, very strong stimulus bringing in lots of positive ions to reach threshold again and to fire another action potential. But once it gets back to resting, it's the same thing. So there are two refractory periods, absolute and relative refractory periods, where you absolutely cannot fire it or it's relatively more difficult to fire it, even though you can. Okay, so this was a very, very quick and dirty summary of this event. There are a lot of other little nitty-gritty details in here, but the key thing is that an action potential is all or none, meaning you're either going to fire an action potential or you are not. And we have this threshold here that determines that. If you hit this threshold, you fire the action potential. It's all or none, and it travels unidirectionally down a neuron. So unidirectionally down neuron, and it doesn't turn it around because of inactivated voltage-gated sodium channels. Then once you reset the resting, you have the ability to then fire another one, or you can fire another one within that relative refractory period. But I hope you enjoyed this little overview on how a neuron works. One thing we're not going into in this course here in survey is synaptic transmission, on how this neuron communicates to the next neuron. That would be in a full A&P course, but here the signal gets to the end of the neuron, releases neurotransmitter, it then communicates and sends that signal, whether it's excitatory, inhibitory, to wherever it's going in the body. In the next video, we're going to start going over all the regions of the brain and how they all work together, along with spinal cord function and things like that. All right. Thank you for watching today. Like always, if you have questions, just let me know, and see you all next time. Bye-bye.